So good morning, students. Today we will discuss about the next topic in this chapter, that is post-fertilization. Post-fertilization, structure in that, events and structure. So in the last class we had discussed about the double fertilization. After double fertilization, what is going to happen? See, in the last class what you studied means that one egg cell, that egg cell will be the haploid, fuses with the one male gamete, that is also haploid, which forms the diploid zygote. Okay, so this is the first fertilization. Second fertilization, in that we had studied the polar nuclei, which is diploid, fuses with one male gamete, which is haploid. It forms a triploid primary endosperm nucleus. Okay, further, during post-fertilization events, means after fertilization, whatever the structures or whatever the events are going to take place, that we are calling it as the post-fertilization events and structures. So, after post-fertilization, these primary endosperm nucleus will develop into the endosperm and zygote will develop into the embryo. Okay, zygote will further develop into the embryo and this primary endosperm nucleus will develop into the endosperm. So here first we will see about the endosperm. See here, the development of endosperm and embryo, here in the post-fertilization, what is the main event is going to take place, means that is the development of embryo and endosperm. The development of endosperm and embryo, here maturation of ovules, next event, that is maturation of ovules into seeds and ovary into fruit. See here, during post-fertilization, the two events are going to take place. First is development of endosperm from primary endosperm nucleus and embryo from zygote. Except this, one more event is going to take place, that is the maturation of ovules. Ovules will mature into the seeds. Always ovules will develop into what? Seeds, and ovary will develop into the fruit. Okay, ovules which are present inside the ovary, so that which is present inside the ovary, that develop into the seed. Okay, and whatever the ovary will be there on ovule, that will develop into the fruit. So we will we will see here how the endosperm embryo is going to develop. In embryo, both dicot embryo and as well as the monocot embryo. Then we will go for the seeds, structure of the seeds, then of fruit. So see, first we will talk about the endosperm development. Here, endosperm. Endosperm. What is endosperm? Let's see what exactly meant by the endosperm. It is the most common nutritive tissue. It is the most common nutritive tissue. Okay. Here, for whom it is providing the nutrition means for developing embryo. Moves for the developing embryo in angiosperm. Means that in flowering plant, what happens means here the endosperm, this will act as an nutritive tissue. What do you mean by this nutritive tissue? Means it's providing a food or the nutrition to whom? To the embryo which is developing. Providing nutrients to whom? To this embryo. Okay, this is the main role of whom? Of the endosperm, which is taking place in all angiosperms or the flowering plant. So, first of all, what is endosperm? Means it is a product of triple fusion. Means after triple fusion only, triple fusion means what? Where the diploid polar nucleus fuses with the haploid male gamete. That process we are calling it as the triple fusion. So after triple fusion, what is going to take place means that is the endosperm. Primary endosperm nucleus. So that is the thing. That's why we are telling that this particular endosperm, it is the product of triple fusion. This is the second thing. Then next here the primary triple fusion and as well as we can also write is the double fertilization. Okay. The next here primary endosperm nucleus PEN. This will undergo into a repeated mitosis division. Undergo into a repeated mitosis. Mitosis means that is the equational division. Mitosis division. For what purpose? It is undergoing into the mitosis division. It's to form or mitosis and develops the endosperm. Or to form the endosperm. See here, primary endosperm will develop into the endosperm. How means here the primary endosperm will repeatedly undergo into the mitosis division. That is the equational division. Then after that repeated mitosis division, this primary endosperm will develop into the endosperm. So here there are three types of endosperm formation. Okay, here in the angiosperms altogether there are three types of endosperm formation. Next we will see how this endosperm is going to form. Okay, that endosperm formation is of three types. That is the nuclear endosperm formation, cellular endosperm formation, and one more, that is the halogeal endosperm formation. So let's see about that one. This is about the endosperm, which is one of the nutritive tissue which develops the embryo. This is the first thing. It is a product of triple fusion and double fertilization. This is the second thing. Third thing, here the primary endosperm nucleus will undergo into which division? Mitosis to form the endosperm. Okay. Next, let's see what are the three types of endosperms. Next up, there are three endosperm, three types of endosperm formation are there, namely endosperm formation are there, namely first one, that is nuclear endosperm formation. Nuclear endosperm formation. Okay, in this what happens? How the primary endosperm nucleus is going to form a nuclear endosperm means. So here the primary endosperm nucleus, whatever the primary endosperm nucleus will be there, that will undergo into the division. Okay, so undergoes into a successive nuclear division, not cellular division. For example, this is the primary endosperm nucleus. Okay, here the primary endosperm nucleus will have the three nuclei because it is a triploid. Okay, this is the cell, that is the primary endosperm nucleus. Inside this, there is a presence of three nucleus. You had studied about the triple fusion. So this nucleus is going to fuse. This is the 1, 2, and 3. This nucleus is going to divide. That means the nuclear division. Primary endosperm nucleus will repeatedly undergo into the what? Nuclear division, not cellular division. If cellular division means total whole cell will divide, but only the nucleus is going to divide to give rise to the, if only nuclei come continuously it is dividing. For example, here there are 3 means in the next stage it will become 6. Okay, in the next stage it will become 12. Like this means continuously it is dividing. Okay, as compared to the first stage, you can see in the later stage many nuclei are going to form. Okay, that many nuclei we can call it as the free nuclei. Many times this nucleus will undergo into the division to form a free nuclei like this. These all are free nuclei. After repeated division of this nucleus of primary endosperm nucleus, it will undergo into the nuclear division to form a many number of nuclei, to which we can call it as the free nuclei. So it may be few thousand nuclei are going to form. Okay, how many? Continuously dividing means 100 also, it may become few thousand. How many? Few thousand of free nuclei are going to form. Okay, so that few thousand nuclei thus produced are pushed towards the periphery. Periphery means at the cortex region of the primary endosperm nucleus, it is going to pushed. How means? I will show you here. See, for example, these are the nucleus. Then later here the nucleus are pushed during the formation of endosperm. This nuclei are pushed towards the periphery, means the peripheral region. This region here I will represent it in the imaginary lines. This is the peripheral region and this is the middle region or the central region. So all these nuclei are pushed towards the peripheral region, like this. Here, I will write free nuclei are pushed towards the periphery. Okay, in this way, all the nucleus is going to push at the peripheral region. Then what happens means pushed towards the periphery of what? Of the embryo sac. So the central part of the embryo sac, thus this central part. See, all over this structure is the embryo sac. As I told you before only, endosperm is providing a nutrients to whom? To the developing embryo. So this is the embryo sac and this is the free nuclei. At the center you can see a large vacuole. What it is? A large space. You can see that space is the vacuole. This is the vacuole. Okay. When vacuole is going to form? When all the nuclei are pushed towards the periphery. Then next one example I will tell you, that is coconut water from tender coconut. Okay, in central whatever the water will be there, that water is the vacuole, that cavity in which the water will be present. Okay, that is the vacuole, and whatever the what we call coconut meat will be there. How will be the tender coconut? Tender coconut. See here, this is the tender coconut. Okay, in center there will be a large vacuole. Okay, that is the coconut water, which is filled with the water. Okay, and all the free nuclei are going to present at the periphery. So this nuclei is going to form a coconut milk. Sorry, coconut meat. That means the white color one structure will be there inside the coconut. That is the coconut meat. Here the coconut meat is formed by the free nuclei, and the vacuole is going to form vacuole. In place of vacuole, what is going to form means that is the coconut water. So one example we can take a tender coconut or the coconut water from tender coconut is the free nuclear endosperm. Here I will mention. Example, coconut water from tender coconut is the free nuclear endosperm. Okay, so in this way, the nuclear endosperm formation will take place. The next, second type, that is the cellular endosperm. So let's see about that one. Now see here, second type, that is the cellular endosperm formation. Here there will be no division of free nuclei, but directly in this case free nuclear division does not exist. Free nuclear division does not exist. Okay, but directly there will be endosperm nuclei is going to followed by the wall formation. Endosperm nuclei is followed by cell wall formation. Here you see, in the last case you had studied only free nuclear division. There will be no cell wall formation, but here there will be cell wall is going to form. For example, this is the primary endosperm nucleus. There directly a cell is going to divide. Okay, cell is going to divide means there will be formation of what? Cell wall in this way. Okay, further it may become this cell wall may divide vertically like this. Okay, so in this way the cellular endosperm is going to form. The first wall is usually transverse. First it will be the transverse, the next, but sometimes it may be vertical or oblique. Example about this is that is the Adoxa. Like Adoxa plant, you can see in this way cell wall formation is going to take place. So this is about what? Cellular endosperm, where there is a cell wall formation after division of the primary endosperm nucleus. It is going to divide the whole cell wall, not only nucleus. Okay, the third that is the fallopian endosperm formation. Fallopian division of nuclear endosperm followed by nuclear endosperm is followed by transverse wall formation. Followed by transverse wall formation. That means first nucleus is going to divide, then the cell wall is going to divide. So here resulting in the form of, see, if first nuclear is going to divide, then the cell wall is going to divide. What is going to happen means here there is a formation of small chalazal cell. Okay, then it will give rise to how means if this is the embryo sac, in this this is the zygote and this is the endosperm nucleus. Okay, this is the zygote and this is the endosperm nucleus. So what happens, sir? I told you zygote will be the like that only, but the endosperm nucleus is going to divide first. How means, sir? This is the zygote, this is the nucleus. of what? Of an endosperm. Nucleus is going to divide, which is followed by the cell wall. Okay, these are the nucleus. This nucleus is divided into two. Okay, as well as it is followed by transverse wall formation. You can see a transverse wall. The next further, it will give rise to the small chalaza cells like this. Zygote will develop into the embryo, and many transverse walls are going to form at the upper region of the embryo sac. That means the chalaza cells are going to form. So this is what? This is about the allovial endosperm. Then endosperm is formed in many angiosperms. Okay. So this endosperms are going to form in majority of the angiosperms. The angiosperms which do not form endosperms are the same. So this is about the allovial endosperm. Next I want to tell you about the endosperm. What is that means? Lastly, endosperm formed in many of the angiosperms. Many angiosperms. Okay, and the angiosperms which do not form the endosperm, they belong to the family. That is, the only angiosperm which do not form endosperm, they belong to the family, do not form endosperm, are the members of the family, are the members of the family. That is, they had given only the family's name. What are that means? Orchidaceae, Orchidaceae. Second one, that it is Podospermaceae. Then third one, that it is Trapeziae. These are the three families in which there will be no formation of endosperm. Except these families, rest all the angiosperms will have the endosperms. Okay. The next endosperm, either it may be consumed by the embryo. See here, first of all, endosperm is helping in the development of embryo. Okay. What happens in some of the angiosperm? The embryo totally utilize the endosperm, but in some it is totally going to consume by whom? By the developing embryo. That examples are pea, groundnut and beans. Okay, let's see how they are going to consume. See here. First of all, endosperm will help in the development of what? Development of embryo. Keep in mind, what is endosperm means? It's providing a nutrients to the developing embryo. This I have told you in first only. But what happens in some of the cases, this embryo is going to totally consume this endosperm. Okay, while it's developing or maturing, okay, at that time this embryo will totally consumed by the embryo. Here there are some examples. That is pea, groundnut and beans. Okay, here in this case, what happens means developing embryo or endosperm may either be consumed by the endosperm consumed by the developing embryo. So these seeds, whatever these seeds you can see in the pea, groundnut, how it will be made. One example I will tell you. This is the groundnut, in which there will be presence of seeds in this way. Okay, these seeds, you can see. See, you see the seed in mango. Okay, how it will be totally enclosed into the certain layers of the fruit. Okay, here in the groundnut, in the pea, in beans, okay, and all you can see the seeds are totally separated from the what we call coat of that. But in pea also you see it will be lying like this, not totally immersed into the fruit. For example, this is the fruit or the covering. So these are the seeds. Here the seeds are totally consumed that endosperm. Okay, so these are the examples. So here this type of seed you can call it as the non-endospermic. The seeds of pea, groundnut, beans and all, you can call it as the non-endospermic because the endosperm is totally utilized by whom? By the embryo. Okay, so some of the other angiosperms in which the embryo will be or the mature seeds, and that mature seed will continue to support the growth of embryo. The endosperm is not going to consume, but it is going to support the growth of embryo during the seed germination. They are such seeds are called as the endospermic seeds. Example, that is the coconut seed and castor seed. The seeds which are endospermic, which are endospermic, that is coconut seed, and one more, that is castor seed. Here in this case, what happens? The embryo is not going to totally consume the endosperm. It will be persistent. What it is, that is the endosperm. Already I told you about the coconut. How the endosperm? It will be in a fleshy meat, to which we are calling it as the coconut meat. There is a persistent of coconut meat. Along with that, one small seed will be there inside the coconut. Okay, so here there is a persistent endosperm as well as the seed. The next, this is about the endosperm formation. Next we will go for the embryo. Okay. Now next, that it is embryo. When the embryo is going to form after in double fertilization, after there is a fusion of one male gamete with one egg cell, there is a formation of zygote. That zygote will develop into the embryo. Okay. So here after fertilization, what happens means, first of all, when embryo is going to form means egg cell that is haploid fuses with the male gamete, which is also haploid. Okay, then it will form a zygote. After fusion of these two cells, it will form a zygote. Zygote will further develop into the embryo. Okay, and all these process we can call it as the syngamy. Syngamy, that is in double fertilization. Okay. Okay, understood. The next here, what happens after fertilization? This zygote, whatever the zygote is going to form, it will undergo into a period of rest. Okay, see here, after fertilization, whatever the zygote will be there, this zygote will undergo into the period of rest. Means it will take rest for some period of time. Then it will grow and gives a size of embryo. Then it will grow and gives a size of embryo. Okay. The next, in earlier stages of embryo development, whatever the stages of embryo development is there, the stages of embryo development is known as it is known as the embryogeny. What it is known as? It is known as the embryogeny. Okay, whatever the stages which is taking place during the embryo development from the zygote, that it is known as the embryogeny. So here there is no much differences between the monocot embryo and the dicot embryo development. It is almost similar, only at a certain point it is different. So here what happens is, actually the zygote will take rest, then it will divide. It will divide into 2 cell, 4 cell, then further it is going to take a shape of an embryo, that is either a dicot embryo or a monocot embryo. I think you know the difference between the dicot and the monocot. Okay, dicot means where there is a presence of 2 cotyledons. Okay, so here in this topic, I have to explain both the embryos, that is dicot embryo as well as the monocot embryo. So that development of embryo I will tell in the next class. Okay, thank you.